Now I'd like to hand over to our first talk. This is Dr. Maria Musaji from Moorfields Eye Hospital. Please give her a welcome.
Good morning, ladies and gentlemen. Good. Okay. Thank you very much for inviting me to speak today. It's an absolute pleasure and the first time I've ever been invited to the Aniridia Network UK conference. I'm a consultant ophthalmologist at Moorfields Eye Hospital and at Great Ormond Street Hospital for Children, and I specialise in genetic eye disease. So if any of you haven't got a genetic diagnosis yet, then I'd be very happy to see you. And if you're being followed up in some of the other clinics in those hospitals, it's likely that you may get referred to see me at some point. I also lead a research group at UCL Institute of Ophthalmology, where my group are trying to understand the disease mechanisms of genetic eye diseases and to develop new treatments. And so hopefully some of my research is going to be moving into the field of aniridia so that we can hopefully make some progress to develop treatments for you in the future. So I was sitting in my clinic about 6-7 months ago, and a gentleman walked in, sat down by my slit lamp, who had aniridia, and I got very excited and I started talking to him about all the research that we were doing and wanting to do, and then he said, "Oh yeah, thanks, yeah, I am quite aware of that. Actually, I'm one of the trustees of Aniridia." And it was James. And so that's actually how my relationship with Aniridia Network UK began, because he very much introduced me to all of you. So thank you very much, James. But I have to say that over the last year, I have come into contact with a number of families affected with aniridia. And I have to say that, you know, the support that you provide your families and the individuals with aniridia, and your drive to understand the condition and to develop potential treatments is really what has driven my work and why I'm here today. And I hope that after this talk, we will have some hope moving forwards into the future. So I am going to be talking about possible drug treatments for aniridia this morning, but before I start, I just wanted to go over how the eye develops normally, and then what really goes wrong when there are genetic mutations that lead to the eye not developing as well as it should. So I don't know if any of you know this, but you see the first signs of the developing eye at around day 21 of pregnancy, really early on, sometimes at a stage where you may not even know that you're pregnant. And the eye grows out from the brain, and it has a stalk, and that stalk becomes the nerve that sends signals from the eye to the brain. And it has a sphere on the end, and as that sphere reaches the surface of the developing embryo or baby, it sends signals to each other. And the surface layer actually forms your cornea, the clear outer layer of your eye, and the lens. And then that sphere actually sort of infolds to form a cup, which actually forms more of the globe of the eye and develops into the retina. And that actually happens within the first 6 to 7 weeks of pregnancy, so really early on. At the same time, a cleft forms at the bottom of the eye to allow blood vessels to grow into the globe of the eye to supply it. And then that cleft fuses again by week 7 of pregnancy. But there are genes that are underway that are signalling the eye to develop in the way that it does at those early stages. And it's the genetic changes in those genes at that time that leads to conditions like aniridia and other developmental eye defects. So developmental eye disorders in total actually account for about 30% of childhood visual impairment and blindness across the world. And it's actually the most common cause of sight impairment in children in the UK. And it includes conditions where the front chamber of the eye hasn't formed, something called anterior segment dysgenesis, which is aniridia is included in that. Cataracts in early childhood, glaucoma, and actual problems with the whole structure of the eyeball, so where the eye hasn't formed at all, a condition called anophthalmia. Children are born with no eyes because the development of the eye is essentially aborted very early on. microphthalmia, where people are born with small eyes that may have other conditions like aniridia associated with it, or problems with the iris causing keyhole defects or clefts extending through the eye. So aniridia, as you're all aware, it's a rare genetic condition. It has a prevalence of around 1 in 40 to 100,000 individuals. 2/3 of aniridia patients have just the condition affecting their eyes only. And these are caused by changes in a gene called PAX6, which I'm sure you've all heard of. And then 1/3 of those patients have something called Wagger syndrome, which means Wilms tumor aniridia genital anomalies retardation syndrome. And that involves 2 genes, the PAX6 gene and a gene called the WT1 gene. And from the pictures that you can see behind me, and you'll all be aware, but aniridia is where you can get a variable difference in the development of the coloured part of your eye, the iris. Sometimes it doesn't form at all, sometimes part of it forms. You can get cataracts, you can get high pressure glaucoma, you can get corneal scarring, and the back of the eye, the retina, there's an area in the central part of your retina called the macula, and an even more central point called the fovea, which is the area that controls your fine central vision, what helps you to see detail and colours. That doesn't develop either. So again, I have a picture behind me which shows what a normal fovea looks like, and you can see a sort of a valley effect, a dip between 2 hills. But in aniridia, you don't get that forming, so you just have a straight plane of the retina. And then obviously if you have Wagger syndrome, you have other problems that affect the rest of your body, like the kidney tumours, learning difficulties, sometimes obesity, pancreatic disorders. The gene PAX6 also can cause other eye conditions. Sometimes they're found in association with the aniridia, sometimes they don't. They just cause other eye conditions on their own. So I mentioned the anophthalmia, where no eye develops at all, microphthalmia, where you get these small malformed eyes, ocular coloboma, this is where you get that cleft in the eye. So where the cleft formed to allow the blood vessels to grow into the eye, it doesn't fuse properly. And so you're left with essentially a cleft in the eyeball. And again, I have a picture on the slides where you see a picture of the back of the eye, the retina, and you have a very small circle. And for those of you who can see, it's that small circle, that is your optic nerve, and you have blood vessels growing out from it. But underneath you have this big white gap, and that's actually the cleft in the eye. That's where the eyeball hasn't fused properly during early development. And PAX6 can also cause nystagmus without any iris problems at all. Patients have the eyes that are moving from side to side, and the fovea, the area at the back of your eye, doesn't develop in those patients. So it's more of a milder clinical features of aniridia potentially. So in the NHS at the moment, there is a big movement towards something called genomic medicine. And this is an emerging medical discipline which we want to integrate into routine NHS care in the next 2 years. And it essentially means we are going to use your genetic code to understand more about you and provide the right clinical care for you. So either for diagnosis or for actually developing personalised treatments for you. And so how do we do this? Well, we have already, over the last few years, through a Department of Health initiative called the 100,000 Genome Project, been taking blood to undertake a screen of every single letter in your genetic code, so all 3 billion letters and all 20,000 genes. And that's called your genome, and it's called whole genome sequencing. And by doing this, it is going to have benefits for aniridia. What are those? So first of all, for those of you who have had genetic tests in the past and had not got a genetic result, it may mean that we will be able to find the genetic cause of your condition. Because traditionally, we used to only look at the instruction parts of genes, specifically the PAX6 gene. But thanks to the great work of Veronica van Heiningen and her colleagues, we have found other genetic mutations outside of those genes where there are special regulatory elements that have been hidden inside other genes that we would normally not pick up on. So we can now, by looking at your whole genome, identify those kind of changes, and so we will increase our diagnostic rates. One of the questions that we often ask ourselves in our clinics when we see families is, why are some patients more severely affected than others, even within the same family? I've certainly seen families where, for example, and I was just talking to Veronica about this, where we had a patient who has the nystagmus, no iris problems at all. She has the foveal, what we call foveal hypoplasia, where the back of the eye hasn't formed properly. And her dad carries the same mutation and is completely normal. So why is that? So if we can look at his genome and compare it to his daughter's, and we can find out what's modifying his genome so that he doesn't have the disease, then potentially can we develop treatments or screen for these things, or at least give you information as to what your future prognosis will be. Again, from looking at your genetics and your genome, we can predict and we can prevent associated disease early on. Okay, so for example, in WAGR, can we find certain genetic mutations or changes that will say that you will develop pancreatic problems or you'll have behavioral problems, and therefore we get the right multidisciplinary team in to work with you early to prevent any future sort of complications developing. Personalised medicine, that's something that I'm going to talk about more, but this is based on your actual genetics and using your genetics to work out what the best treatments are. And that follows on to a very long word called pharmacogenomics. So what does that mean? So basically, it's using your genetic code to determine what drugs will be safe and effective for you. Currently, we spend over $300 billion a year on drugs that do not work on patients. So by using a patient's genomics, we can now work out, as I just said, the right drug that is going to have a positive impact on you. So one example of the work that you would have heard of over the past few years, but an area that I also have worked with Dr. Cheryl Gregory Evans, because I did my PhD with her at Imperial College between 2005 and 2008, is nonsense suppression therapy. Now, there are many genetic changes that can cause disease. And there is one particular type of mutation called a nonsense mutation that causes up to 70% of human genetic disease. Now, a nonsense mutation is where a single letter in a gene changes and it introduces an abnormal stop signal within that gene. So when your protein-making machinery is reading the gene and it sees that abnormal stop signal, it just stops. And so it doesn't produce normal full-length protein that can work, and that's what essentially leads to the disease. Now, in aniridia, about 40% of the mutations are nonsense mutations. And now there is a class of drugs that can be used to override these abnormal stop signals, and it works in a disease and gene independent manner. That means it doesn't matter what disease you have, what your clinical features are, or what gene causes it, but if you have a nonsense mutation, then this drug may be helpful for you. So the diagram behind me, again, it shows how the drug works and what happens normally within our cells. So we have our gene, which has, it provides the instructions of how to produce the protein. And the protein making machinery go along reading these instructions and they produce the normal full length functioning protein. In the presence of that abnormal stop signal, when it hits that stop signal, it stops making protein and you're left with this non-functional protein. But in the presence of the drug, it can override that stop signal, and it can lead to the production of full-length functioning protein. And it produces about 25% of working protein, which is enough in a lot of conditions to potentially either halt or slow down disease. This is not a drug that can reverse disease. So if cells are dead or cannot be regenerated, then it won't work. But if there are cells that are even sick or unhealthy but still alive, then by producing a baseline level of some protein will help increase its function and its health. And there are several drugs that have been developed, but one of the first ones, which is what this work was all based on, was on a class of antibiotics. So these antibiotics we currently still do use in our hospitals to treat infections. And the way that they work on bacteria, it works on a milder way on our human cells. So what I did back in my PhD was I had a zebrafish model of a zebrafish that had a cleft through its eye. It had a small microphthalmic eye, and it also had some kidney defects, and it was caused by a mutation in a gene called PAX2. And it causes in humans something called renal coloboma syndrome. And so I tested this class of antibiotic drugs on these zebrafish, and what we found was that it actually managed to fuse the cleft in the eye, and the fish itself looked healthier, and it nearly doubled its lifespan. So we felt that these drugs had a really good effect, and we used these on various other different disease models with similar effects. But the problem is that these antibiotics are fine to be used for a very short course to treat infection. But if you're thinking about giving them over a long period of time, they actually have significant side effects. So they're known to cause hearing loss and kidney failure. So we sort of hit a bit of a roadblock because we didn't know what to do. We couldn't translate it any further to patients. So then over the coming years, we worked with medicinal chemists to design new molecules where we chopped off the bits that caused the side effects, but we kept the bits that were actually functioning to override these stop mutations. And there were also high throughput drug screens that were undertaken by companies such as PTC Therapeutics. And they identified the drug called Ataluren. And so there was a few years sort of in between this, and this is where Cheryl Gregory Evans then moved to Vancouver, which is where she's based now, and she did a study where she gave the new drug Ataluren to the Aniridia mouse model. And what she did was an interesting thing. She gave the drug to the mice after they were born, in the 4th day of life. And she gave the drug for about 10 days, and then she looked at the eyes again. And what she found was something that was, for me, quite remarkable, that the developmental defect had almost been reversed, and that the eye looked much more like a normal eye. And we're not quite sure if that actually works with humans, because I've given you the picture that in humans, the eye develops very early on, within the first few weeks of pregnancy. However, the back of the eye, the fovea, that develops in the first year of life in humans. So potentially, if you gave this drug very early on, you could actually enhance the remodeling of the back of the eye once the baby's born, which would actually potentially improve the visual function of a child. And then, because you're giving the drug orally, it could actually then help to support the cells of the cornea, the iris. What I wouldn't expect to see is that the iris regrows once you start to give this drug in humans. But nonetheless, this is the results that they found. So Ataluren, the drug, there is now a large body of evidence that it works in a number of animal and human cell disease models. It's actually gone through phase 3 clinical trials for cystic fibrosis and Duchenne's muscular dystrophy. It is known to be safe and tolerable in children as young as 2 years onwards and in adults. It's a powder that you dissolve into water and drink 3 times a day, and it has minimal side effects. So the company has now treated over 1,000 people with over 5 to 8 years of follow-up data, and there has never been a serious adverse event or any ocular adverse events. What they have found is that sometimes in some individuals they complain of nausea and some transient diarrhea when they first start taking the drug. So overall, this compound is safe to use. It's been given approval by NICE in the UK for the treatment of Duchenne's muscular dystrophy, and I've just put down here some of the reasons why it was given approval. Because when, for example, if Ataluren is found to be successful for aniridia, it's important to know the kind of canvassing that Aniridia Network UK and other patients may need to convince NICE to approve such an expensive treatment. So it was found that none of the children that were taking the drug lost the ability to walk over a 48-week period of the trial, compared to 8% of patients who were taking a placebo or the non-drug version. It was predicted through modelling that Ataluren may delay the loss of walking for up to 7 years, and patient experts said that they had seen a meaningful stabilisation or an improvement in their child's mobility, such as being able to get out of bed independently or go to school. And they pitched that if a child lost the ability to walk, there was a greater deterioration in them being able to conduct activities of daily living like self-feeding, showering, going to the toilet. And being able to walk for a little bit longer would allow them the retention of normal adolescence. And so they approved the treatment, and the treatment costs around £220,000 per year. So, as you all know, Ataluren has gone into a phase 2 clinical trial for PAX6 nonsense aniridia in the US and Canada under the Gregory Evans. It's a randomized double-masked placebo-controlled study. So it involves half of the patients taking the drug for 48 weeks and the other half of the patients taking the placebo, and then both groups are allowed to take the drug for a further 48 weeks. It's, as I said, an oral dose taken 3 times a day. The primary outcome of this trial is safety, because it has never been given for an eye condition indication before. And the secondary outcomes are looking at the visual acuity, how well the patients can see, the cornea surface, and measuring the iris area. And I contacted Cheryl for an update, and she said that all 40 patients had been recruited and they were currently being followed. But the end date of the trial is December 2019, so we wouldn't really hear any results until early 2020. So still, unfortunately, quite a while. However, I have said that if the results of this trial are positive, then I would be very willing for Moorfields to be a site for a phase 3 clinical trial. So I will keep you updated on that. So in the meantime, there have been a number of other drugs that we have been testing in our group. So the ones that I said the chemists were working on to see how well we could modify chemicals, etc. And this is where we move to the research part. So we're still in sort of the preclinical field for this. We're trying to build up the evidence so that we can translate these to our patients. But we've been looking at a number of molecules, and I use the zebrafish as a quick screen to see if these molecules are safe or not. And one of the first things we do is we take normal, healthy zebrafish and we give them a range of doses to find out the safe dose, and then we look further to see, is it having any sort of damage on the kidneys, and we can automatically identify those where, for example, there are signs of kidney failure after you dose the fish with this. We can also do hearing screens. So the zebrafish have hair cells that not only help them hear but sense vibration, and we can use the original drugs that cause the toxicity and compare them to the newer compounds again to see which one of those are of benefit to pursue as potential candidates. And then we can look at the zebrafish eye. We can take cut sections through the eye, looking for restoration of anatomy, to look for levels of cells dying, and we can look at oxidative stress. We can do a number of things. But essentially, in all of the candidates that we tested, the best one that was coming through was ataluren. So that's promising for all of us because we know that drug is in clinical trials. But there is a new candidate on its way which also has a lot of benefit that at least we have found in our laboratory to have a similar effect to ataluren, and that's a drug called amlexanox. Now this drug was found through a high throughput drug screen. It's already an approved drug in humans, so it has been used for over 20 years to treat asthma and mouth ulcers. It's already been tested on cystic fibrosis patient cells, and what we're hoping to do in the next 2 years is test this on aniridia disease models to see if we can now translate that to a clinical trial, because we won't have to worry about all the safety studies because we know it's safe to use in humans, and we can essentially repurpose this for aniridia. So amlexanox, I'm going to add a slightly extra layer of complexity to what I'm talking to you about. So the way ataluren works, as I said, is it just overrides that stop signal and produces functional protein. But our cells are very clever things, and it has a natural surveillance mechanism within our cells. So if we have a gene that has a nonsense mutation, when it produces the instructions which has that mutation in it, our cells degrade it and get rid of it, because what it's trying to do is protect you. It doesn't want a build-up of these sort of toxic proteins potentially. So as a result, it ends up chewing up and degrading all the what we call the substrate, the food for the drug, because the drug needs those instructions to generate protein. So what amlexanox does is it prevents that natural surveillance mechanism, and it increases the instructions that are floating around in the cell, so it has more of that to then read through the stop mutation to make more protein. And we have certainly found in other disease models, inherited retinal diseases, that it increases the number of gene instructions within a cell, but it can also produce more protein, and equivalent, and in some cases slightly more than ataluren. So for patients, this is coming back to why we want to know use genomic medicine and look at your genome, is that we can now measure how well your natural surveillance mechanism works in your cells, and if it's working really well and it's reducing the number of PAX6 gene transcripts, then we may potentially in the future opt to give you something like amlexanox. Whereas if you had a patient who the natural surveillance mechanism wasn't working so well, then we would just need to give you ataluren. But it also gives us the choice as doctors, and if one stops working, we could try a different one. And it's always good to have competing compounds because it drives down the cost as well. So one of the models that we are now going to be using is a stem cell model of an early developing eye. So incredibly, we can now take a skin sample from a patient, we can convert their skin cells into stem cells, and then we can grow an early eye cup from those stem cells. And again, the picture on the slide shows initially a ball of stem cells, and then they've been converted into early eye cups, and they are those floating spheres in that corner. And then when you zoom up and you take actually a section through one of those developing eye cups, you can see the early developing retinal layers. And all the red staining behind me on the bottom of one of those cut sections is the PAX6 protein. And that's seen very early on in eye development. So what we're going to do is we're going to actually be taking a skin sample from a couple of patients, growing their eye cups. We shouldn't see any PAX6 in those cells. We may be able to see how it affects development, learn more about the molecular mechanisms. But if we give those eye cups the drugs and they're working and they start to produce PAX6 protein, we will be able to see and measure that. And that's what we're hoping to do. We can also continue to monitor those developing eye cups to see how well the other layers of the retina form. And we now, the picture again behind me is of a week 20 eye cup, where we actually begin to see early light sensing cells, the photoreceptors developing in the retina. And we know that PAX6 is involved in various signaling of differentiation of certain cell layers. And so we'll try to compare normal with aniridia eye cups and then treated eye cups to see what the benefits are. The other model that we are potentially looking at using for high throughput drug screening is the aniridia zebrafish model. And the picture behind me just shows what the obvious difference is in the eye. So this is, if you can imagine, you're looking at the zebrafish from the top down, and the zebrafish eyes are a bit boggly and they stick out from the side of their head. And you can clearly see the difference between a normal and aniridia eye. And so what we're hoping to do is high throughput drug screens of known approved drug libraries to see if we can see a difference. And then we can pick up those candidates and we can use those to test them on our special eye cups that we've grown to get more detailed information. And because these are approved drugs, if we do find a potential drug candidate, we can then move swiftly on to clinical trials with patients. So, how can you all get involved? So, first of all, if you don't have a genetic diagnosis yet, please do either get a referral from your GP to see me. I'd be happy to undertake genetic testing for you. Or if you're already seen at Moorfields, then just send me an email. I'd be very happy to find out what's going on and facilitate that. Knowing your genetic diagnosis helps us to develop treatments. It helps us to identify targets that we can then develop therapies for. It will obviously help you for genetic counselling and potentially for family planning. And it helps us to get the right multidisciplinary team to support your care. The other thing that we are very keen to do, and again I was speaking to Veronica, the patron, earlier, is that we don't quite understand the relationship between certain genetic mutations and the clinical features. So in the coming years, we would like to start putting together what we call a genotype-phenotype study. That means looking at your genetics and the clinical features and trying to work out why some people are affected more severely than others. Because again, that will all help us towards developing treatments and understanding the disease. And the other reason for investigating your clinical features carefully over fixed periods of time is to find outcome measures for trials. Because if we don't find the right outcome measure, then it doesn't matter what trial or drug or gene therapy we develop, it may fail because we haven't got it right. And that may be questionable with the aniridia study as well that's ongoing. Measuring the iris size, I think, is questionable. I think there were probably better parameters to measure, but at the moment we're not quite sure what they are. So these are the reasons why, if you want to get involved, then I would be very happy to receive an email, even just expressing your interest. For you to get more information, you can access something called www.clinicaltrials.gov. That is a website that lists all approved trials internationally that I would say have got ethical approval. It's run by the National Eye Institute in the US. And so if you go to that website, it says search studies. You just type in aniridia or PAX6 or WT1, and it will come up with all the studies that are related to your condition. And then the last thing is obviously fundraising, because although we want to do all these things, sadly we can't do it without support. So I was very pleased that Aniridia Network UK joined in the partnership with Fight for Sight recently to put forward a small grant, and I certainly have applied for that, so yeah, fingers crossed. But you know, that's the kind of thing we need to resource these studies. It's actually just manpower so that we can push forward. We have you, we have the data, we just need that kind of support. So to conclude, Phase 2 clinical trial for Ataluren is underway. The results are due in 2020. We are developing patient-derived PAX6 aniridia models to further our understanding, both molecularly, structurally, and to develop new treatments. We're screening approved drugs so that we can repurpose them and for faster translation to you guys. with a particular focus on Amlexanox as a very strong candidate. And please get involved, support the research. Treatments are becoming a reality. The first gene therapy for an inherited retinal disease was approved in the US just in December. There are many clinical trials underway for gene therapies. There are other genetic therapies. We can look for common pathways. So yeah, please support us, and we look forward to updating you in the future. I'd just like to thank my group, my funders, and Aniridia Network UK. And I've left my email address. So for those of you who can't read that, it's maria.mooserjee@moorfields.nhs.uk. But I will make sure that James can send that out to all of you as well. Thank you very much.